Welcome back. So last video I talked about the cardiac cycle and blood and things like that. Today we're going to kind of keep going on that. So I talked about, last video I did talk about the volume curve of like a single cardiac cycle. And I talked about inotropy and all that thing, and afterload and preload. So we're going to talk a little bit about a lot of different things. So I kind of want to start out with a graph. I know I start all my videos out with a graph, but it's something that can maybe help us understand what's going on. So we have a pressure time graph. So pressure time graph, and the pressure in millimeters of mercury are going on right here. So we kind of have, right here, we have a little bit of a, and so I'm going to describe in a different color what's going on in each of these sections. Because as each section goes on, pressure changes, right? So what I mean by each section is if we took a molecule of water, if we were a red blood cell, and we're taking this molecule of red blood cell, and if we, for example, just followed, if we entered the magic school bus and we just followed that red blood cell, where is it going, and what's the pressure at that stage? So right here is actually like the aorta, right? It's high up in pressure, and it depends whether we're systolic or diastolic. Remember my last video I just discussed systolic and diastolic. But anyway, as time persists, we kind of see this kind of go on. Kind of decreases a little bit, and it kind of just keeps decreasing. It never really goes to zero, but it goes, it decreases a lot. And we have, I'm going to draw this in red. We kind of have this average line that we could make. And this average line is just mean arterial pressure, right? The mean arterial pressure. And notice that it is closer to diastolic pressure because we spend more time not pumping than we do pumping or whatever. And we'll put this at 90. Then eventually you go down, right? I said the aorta is right here, or large arteries are right here. We go down as time persists. We're still that red blood cell. Time keeps going on. Even though this time isn't super long, it's in a matter of seconds, we do at some point hit capillaries. So kind of at the 30 mark, right here, capillaries. And then eventually we hit veins. So this is very important. So you're like, oh, okay, so capillaries have more pressure than veins, and that is correct. But we also need to understand that arteries, or large arteries like the aorta, have larger pressure. So again, I kind of want you to imagine this. This is kind of weird to imagine, but if you took all the arteries and veins in the body, and you somehow took them out and you drained them all, and somehow you added blood to the heart and started pumping the heart. As the heart pumps, the blood would start in the arteries, obviously, because arteries take stuff away from the heart, takes the oxygen, the fresh oxygenated blood from the heart, and delivers it to wherever it needs to go. Whether that's a muscle in the arm or a muscle in the leg, it doesn't matter. The blood gets there from arteries, and because it's oxygenated blood. And veins take blood, not good blood, from the muscles and stuff back to the heart, so then that blood could get oxygenated and things. So we have the pressure going on through here, and so the capillaries kind of, I wouldn't say it's a mean pressure or anything, but we have most of the pressure from the capillaries coming from the arteries, and that's probably the main point I need you to get before I start teaching about capillaries in the next phase. And then you might be like, well, so if veins are so low, how do they get back to the heart? Like, if there's blood in my feet and I'm 6 foot tall, how does that blood go from my feet to my heart? And that is the musculovenous pump. Your muscles are constantly contracting to help push that blood up. Okay, so let me now explain what's going on with I'll explain capillaries, but I want to actually explain these things called baroreceptors first. I think that will actually help us a little bit with capillaries. Just please remember this as I teach baroreceptors. Okay, so baroreceptors are very cool, I think. And it's just this thing that's going on all the time within our bodies. And they don't like to get stretched, but they'll get stretched. Again, this is not anatomically correct or anything, but blood is getting pumped from the heart here, right? And it's going up throughout the whole body. And these are the carotid arteries. Again, it's not anatomically correct at all, but these are the carotid arteries, and this is the carotid sinus. And we have these things that I'll draw in green that are called baroreceptors. And they're very sensitive, super sensitive. So when blood pressure rises, or just the pressure of everything rises, these baroreceptors stretches. And when that happens, the baroreceptor stretches, it has an afferent pathway to the brainstem. And then the brainstem will use the autonomic nervous system to go and activate alpha-1 receptors. Yeah. And so as alpha-1 receptors are activated, a lot of different things happen, right? As you know from my previous videos. When I actually want to explain, alpha-1 receptors are very important to radius. And so remember I talked about MAP and how MAP, mean arterial pressure, is equal to peripheral resistance and stroke volume and cardiac. I explained that whole thing. So we could have this new equation actually. So we'll talk about peripheral resistance. So this part doesn't really matter because like the volume and things they do change, but not like a crazy amount over time. It like takes a long time for it to change. And so if we're doing like a procedural diagnostic example of things, we don't really need to pay attention to these, and these could be more constant. And so really, because pi is also just a number, this is the only thing that changes, the radius. And the radius of these arteries, right, we could remember, hopefully maybe you've taken some anatomy, that there's muscles around them. And those are activated actually by the alpha-1 receptors. And so you can kind of see this correlation right here between what's going on. So maybe we need to contract, maybe we need to dilate the arteries so that this pressure could kind of go down a little bit. We could also see cardiac output. There's an equation for cardiac output. I'll put it right here. Cardiac output equals the change in pressure. Again, cardiac output times peripheral resistance. We have things going on. So that is what's going on with the body. And there's so many other things going on, like with the RAS system in the kidneys. The RAS system could get activated in the kidneys, and we could have eventually angiotensin 1 turn into angiotensin 2 from the ACE enzyme. Angiotensin 2 will then increase things like vasoconstriction or thirst even, and adenosine. So there's a lot of different things that go on with it, but I'm going to now explain capillaries. Okay, so again, I want you to remember that graph. In that graph, we saw that the pressure in arteries are much greater than the pressure in capillaries, which are much greater than the pressure in veins. Kind of remember that a little bit. We could even kind of draw a thing right here, like the pressure in arteries is greater than the pressure in capillary, which is greater than the pressure in veins. And so let's just draw the capillary. And you might know what a capillary is, you might not know what a capillary is. Mainly, the capillary is from when an artery turns into a vein. So you need to remember the rule. Artery is taking stuff from the heart, or taking blood from the heart to the stuff, and veins are taking the stuff, or taking blood from the stuff to the heart. So arteries, blood to the stuff, veins, blood from the stuff to the heart. So yeah, we'll have artery side. We'll draw the artery side in red. And then we'll draw the venous side in blue. So venous side, atrial side. And then blue and red mix and they make purple. So there's a couple things that we need to understand here. Again, if we kind of looked at the body and we saw that if we took everything out, or if we had the magic school bus and we went into a red blood cell, starts in the heart, right when we get the oxygen from the lungs, it starts in the heart and then goes to the place. So that's where we're starting the capillaries. We're starting the arteries because blood starts there. So blood's moving into the arteries. As blood moves into the arteries, we have different things happen. So there's a couple different things that I could start out with. There's this thing that I really want to talk about, and it's called albumin. Albumin is a molecule that is a big molecule, and that's pretty much all we really need to know about it right now, is that it is a big molecule. And albumin, I'll draw that right here. Albumin is too big because we have, in every situation or in every circumstance that I've drawn in this video, or in my previous videos in the channel and things, we have channels. And so, same thing with the capillaries. We have these channels in the capillaries that move stuff across. But albumin is too big to go through this capillary. So, what does albumin do then? It creates a concentration gradient. Now, concentration gradient is important for what? Osmosis. So, we also have muscles right here. And these muscles, because this is a generic diagram, could be any muscle. Is this your rectus femoris? Is this your biceps brachii long head? Is this your sartorius? I don't know. Is this your pectoralis minor? I don't know. These represent muscles. And muscles, as most people know, you might not know, are at high concentration of protein. A pen sucks. High concentration of protein. So we have this blood coming in, but we also have this thing on the capillary. Kind of wraps around the capillary like this, over and over, like it's just this big thing. I'm oh, that's kind of cool. Yeah, it is. That is called the pre-capillary sphincter. Sphincter can squeeze, right? It's a muscle. It squeezes, so it might increase pressure and decrease volume, whatever it might be. So it changes the thing. So if you know any sort of physics or anything, we talk about pressure, and there's this thing called hydrostatic pressure because these are water based things. So in the arterial end, we have a hydrostatic pressure of 30 millimeters of mercury. And on the venous end, we have a hydrostatic pressure of 10 millimeters of mercury. And you're like, oh, Jackson, what in the crap are these numbers? And I'm like, I actually don't 100% know how they found those numbers. You could look it up. Google's a thing. But those are just like the normal numbers of hydrostatic pressure for the atrial side and the venous side. So you can see now that graph kind of makes sense. We have a higher pressure in the atria than we do in the venous side, or the arterial side, sorry. But this is the capillary, so it's a little different. It's changed a little bit. So as we have this hydrostatic pressure come in, hydrostatic pressure actually starts pushing solutes out of the capillaries. And we need to understand as well, so the muscles are full of protein, but also is the interstitial space just full of protein. These green squiggly lines represent a protein. Just full of protein with an interstitial space. And as the interstitial space is full of protein, right, there's different concentrations. Protein also is too big to fit through this. So the only things that could do it is just different types of solutes, like water. And we're going to talk about water right now. So because of this albumin. This albumin actually creates an osmosis concentration gradient. Remember in the first video that I taught, we talked about concentration gradients. And so it actually draws water from the interstitial space into the just into the capillary. And so we kind of have this change in concentration gradients and things. And so as these muscles contract and things, we have a couple things leave the muscles. And what leaves the muscles is lactate, hydrogen ions, potassium ions, and carbon dioxide. These all leave. And you might be like, oh, okay, so that's what causes soreness. No, I'm sorry. Maybe like a difference in hydrogen concentrations, maybe, but not lactic acid. I was taught that incorrectly. It is incorrect. It has been falsified since the 1980s. Lactic acid, it's not the soreness. You don't need to stretch it out or whatever because it goes through really fast. But what I need to actually discuss is lactate, hydrogen ions, potassium ions, and carbon dioxide. They don't go to waste. The body is very, very, very good at not putting stuff to waste. And so the precapillary sphincter actually has receptors for each of these things. And so they'll go and connect to these receptors. And once lactate or a hydrogen ion or potassium ion or carbon dioxide attaches to one of these receptors, it actually opens the precapillary sphincter more, which opens up the capillary, more blood could come in. So that's what's going on here. So we have this thing called BCOP and ICOP. So BCOP and so we could actually measure BCOP and ICOP from different things, and dependent on your heart rate, dependent on mean arterial pressure, right? As mean arterial pressure increases, which I'll put right here, because this is the atrial side or arterial side, can change a lot of these things. It could change hydrostatic pressure, it could change BCOP, it could change ICOP. So actually, because ICOP depend on the pressures, so we might have a greater pressure. If we have a greater pressure in the capillaries, more stuff's going to want to come out. If we have a greater pressure in the interstitial space, more stuff's going to want to go in. So it depends on BCOP or ICOP. And so actually, like, water can move out due to ICOP. So ICOP will kind of suck it out and stuff. But we have these other things, because you're like, well, if water's coming out, like, where in the world does it go? That would be a very good question. But we have this thing. We have lymph vessels. These lymph vessels have a negative pressure, like a vacuum. Vacuums have negative pressure. It creates a difference of pressure, so it could suck things up. Lymph vessels do the exact same thing. So they suck up solutes, and so they can suck up water. So now we could talk about a little bit of clinical things. So depending on the blood osmotic pressure or the capillaries or whatever, and it depends on what, like, we could literally zoom in right here, and BCOP and ICOP could be different than it is right here or right here. It just depends. And so we can calculate these things, just calculate what's going out, what's going in. And so, I'm trying to think where could I draw this. But I like to draw it like this. This is a lot easier than trying to draw negatives and positives and trying to flip the negatives and the positives. So you just have to think of what's going in and what's going out. So BCOP in a normal person is 28, and ICOP normally is 8. And then we have hydrostatic pressure right here, and this is just the capillary. We have hydrostatic pressure, and that's 30 because of here, and P, which is equals 3. So we could have this equals 41, this equals 28. The difference between 41 and 28 is 13. But it's favoring this side. This side's bigger, so it's out. So we have 13 millimeters of mercury coming out. So I'm sorry, this looks horrid. It's a mess. But you might ask, so Jackson, like, what if we have too many things coming out? And that is a very, very, very good question. And that is called edema. And edema is when there's just abnormally amount of stuff. There's not a normal amount of stuff in the interstitial space. There's too much. So this can cause a lot of different things. And remember I talked about renin and I talked about kidneys and things like that. And so sometimes we do, if someone's only consuming carbohydrates and they were starved before and they're only eating carbohydrates, we kind of see sometimes a bloated abdomen and a bloated ankles because the interstitial space actually, ICOP pulls a lot of this stuff out. The lymph nodes don't work as much. Water comes out into the interstitial space, and due to gravity, it pulls it down, right? We could also think if mean arterial pressure, so as exercise happens, mean arterial pressure goes up. That makes sense, right? Like your heart's pumping more, your body needs more oxygenated blood, so it just pumps through stuff more. So we can also see, maybe you understand what's going on with gym stuff. I don't that much, obviously, but we have a huge gym culture right now, and a lot of people like to see their pump, right? They like to pump and then look at themselves in the mirror and see how swole they are. But that only lasts for a little bit. And you might ask, like, what exactly is happening there? And that literally is because mean arterial pressure is rising. And so if mean arterial pressure actually is above 160, then that's what's kind of happening. We have all those compartments within the muscles and things, and those compartments can fill with more. So we have more edema, pretty much. That's what edema is. And yeah, that's pretty much all I really wanted to explain. And so sometimes, there's a lot of other stuff I like to explain. So like I like to run. And for the longest time when I started running, I had like a pain in my side. And that is very common actually with a lot of people. And you might be like, okay, what in the world's going on with that pain? Why do I have this pain here? Like, should I go to the hospital? Like, things like that. Usually you're fine. But I'm not a doctor, so don't take my word for it. So what actually happens is when we start running, we actually release more norepinephrine. As we release norepinephrine, that activates alpha-1 receptors. Remember, alpha-1 receptors are very important with all these things. As alpha-1 receptors go throughout the body, this actually opens up channels within the capillaries or potassium channels, and blood will start going there, right? That's the main idea. Blood will start going there. So actually what will happen is because your legs need so much blood, and this needs to happen so much, the exchange needs to happen because your muscles need that oxygenated blood, it will actually shut off some of the capillaries and those things, and because there's no blood getting there, you kind of have a pain there. And so that's why usually when you stop, it stops, because your legs need less blood. Blood could actually finally go there. But edema is just mainly swelling, and yeah, that's pretty much what I wanted to discuss today. If you have any questions, there were a lot of questions last time. I answered them all, whatever. But if you have any questions, please let me know. But I'll see you next time.